Hello everyone, I am Dr. Sirajul Faizi. I welcome you in my YouTube channel. Today in this video, I am going to talk about the chemistry of carbon. You know, the atomic number of carbon is 6. The symbol of carbon C. The element's name carbon and the atomic weight of carbon is 12.01. You can see carbon contains 6 neutron, 6 proton and 6 electron. So look at the structure: methane, isooctane, butadiene, acetylene and benzene. So the uniqueness and the variety of carbon, how carbon arranged in different compounds in different way. Now you can see the nucleic acid, adenine and guanine. Adenine, you can see the phosphate group, sugar, ribose and base. In guanine, see the phosphate group, sugar, ribose and base. Now glucose and fructose. You can see the other form of carbon, N-azido dimethylamine, N-azido N-methyl acetamide, N-azido N-methyl aniline, N-azido acetanilide, N-azido diphenylamine. Now look at the different form of carbon. Look at this molecule. It is very familiar compounds called methane. Don't forget the structure and function relationship. The shape of molecule is important because the structure often determines function. Or if you prefer, the shape probably evolved for a particular function. You can see the 2 normal bond and 2 special bond, one towards you, one behind you. Now you can see the chemical groups: alkane, alkene, alkyne, phenyl, ether, alcohol, amine, alkyl halide, aldehyde, ketone, carboxylic acid and esters, amide. Now you look at the functional groups take place in the chemical reactions: hydroxyl group, carbonyl group, carboxyl group, amino group, sulfohydryl group or thia. Now macromolecules. You know carbon exists in macromolecules. Organic molecules join together, a smaller organic molecule join together to form larger molecules, macromolecules. 4 major classes of macromolecules like carbohydrate, lipids, protein and nucleic acid. Look at here the polymers. Long molecules built by linking a chain of repeating a smaller unit together. Polymers, monomers, repeated small units and held together by covalent bond, shared pair of electrons. Look at here short polymers, unlike unlinked monomers, and you can see the longer polymers. So how to build a polymers? Condensation reactions, dehydration synthesis, joint monomers by taking H2O out. One monomer provides OH, other monomers provides H. Together these form H2O and requires energy and enzymes. Look at here. So how to break down the polymers? Hydrolysis. Use water to break apart monomers. Reverse of condensation reaction. H2O is split into hydrogen and OH. H and OH groups attach where the covalent bond used to be, and hydrolysis is used in the digestions to break down large macromolecules. So carbohydrate. So carbohydrates are composed of carbon, hydrogen and oxygen. Carbohydrate means carbo, carbon, hydrate, ATE. You can see the CH2O. CH2OX, example C6H12O6. Energy is a functions, raw materials, monomer, simple sugar, monosaccharides, example sugar and starch. This is also act as the energy storage in the structural materials. Look at here the structure. Now, what functional groups? Carbonyl, aldehyde, ketone, and hydroxyl. Now, sugar. Most name for sugar end is OSE, classified by number of carbon. 6 carbon hexose, 5 carbon pentose, and 3 carbon triose. So 5 carbon and 6 carbon sugar form ring in aqueous solution in cells. So notice carbon are numbered. How you will number this? So numbering of carbon 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, 6 prime, and oxygen. So simple and complex sugar. Monosaccharide, simple 1 monomer sugar glucose, disaccharide 2 monomer sucrose, polysaccharide large polymers starch. So look at glucose, fructose and sucrose structure. Disaccharide formed by dehydration, synthesis, and 2 monosaccharide joined by a glycosidic linkage. Now building sugars. How can you build the sugars? Dehydration, synthesis. Monosaccharide, glucose, glucose, you will get the maltose, glycosidic linkage. Monosaccharide, glucose, fructose, you will get the sucrose, glycosidic linkage. This is disaccharide. Now polysaccharide, polymers of sugar cost little energy to build, easily reversible, release energy, function, energy storage, starch, glycogens, building materials, structure, cellulose, plants, and chitin, arthropods, and fungi. So branch versus linear polysaccharides. Look at the chloroplast and starch. Amylose, amylopectin, starch. And glycogen, look at the mitochondrion, glycogen granules. So polysaccharide diversity, the isomers of glucose, how does structure influence functions? So molecular structure determine function. You can see the alpha glucose, you can see the beta glucose, only changing the position of OH groups. So digestion starch versus cellulose. Starch 1,4 linkage of alpha glucose monomers and cellulose 1,4 linkage of beta glucose monomers. So cellulose, most abundant organic compounds on earth. You can see the cell wall, plant cell, microfibril, and beta glucose monomers, cellulose molecules, cellulose microbills in plants cell wall. Now glycemic index, the ranking of carbohydrate based on their immediate effect on food glucose, blood sugars levels. Carbohydrate foods that break down quickly during the digestion have the highest glycemic indices. Their blood sugar response is fast and high. So glycemic index, which food will get into your blood more quickly? Apple 36, rice cakes 82, corn flakes 84, bagels 72, peanuts 33. Now lipids. Lipids are composed of carbon, hydrogen, oxygen, long hydrocarbon chain, diverse group fats, phospholipids, steroids do not form in polymers. Big molecules made of subunit smaller molecules, not a continuing chain. Look at here. Fats, structure glycerol 3 carbon plus fatty acid. Fatty acid long hydrocarbon tails with COOH group at the end. Look at the palmitic acid, fatty acid. And look at the glycerol, dehydration synthesis. So triglycerols, 3 fatty acid linked to glycerols, and ester linkage between OH and COOH. You can see the triglycerols, fat molecules. Now dehydration synthesis, you can see the fat is palmitic acid. Dehydration synthesis, ester linkage. Now long hydrocarbon chain, polar or non-polar, hydrophilic or hydrophobic, functions like energy storage, very rich, 2 into carbohydrates, cushions or organs, insulates body, and think whale bubbler. So all carbon bonded to hydrogen, no carbon-carbon double bonds, long state chain, most animal fats, solids at room temperature contribute to cardiovascular disease and plaques deposits. Look at here the saturated fats, stearic acid. Now, carbon-carbon double bond in the fatty acid, plants and fish, fat, vegetable oil, liquid at room temperature. The links made by double bonded carbon prevent the molecules from packing tightly together. You can see the oleic acid. Double bonds causes bending, unsaturated fat and fatty acid. Now, phospholipids. Glycerol plus 2 fatty acids plus phosphate. Phosphate negatively charged and other small molecules may also be attached. Adenine, ATP, adenosine triphosphate. So hydrophobic or hydrophilic. Fatty acid tails, hydrophobic. Phosphate, hydrophilic head. Dual personality. Interactions with the H2O is complex and very important. You can see the phospholipids. Choline, phosphate, glycerol, fatty acid, one linear, one bent. Now, phospholipids in water. Hydrophilic heads attract to water and hydrophobic tails hide from water. Self-assembly auto aggregates, micelles, liposomes, early evolutionary stage of cell. You can look at here the water hydrophilic head and hydrophobic tails, water bilayer liposome. So why is this important? Phospholipids defined outside versus inside. Where do we find phospholipids in cells? Cells membrane. You can see the water, you can see the phospholipid membrane, you can see the bottom layer of water, hydrophobic and hydrophilic. So outside of cell, inside of cell. So phospholipids of cell membrane, double layer, bilayer, hydrophilic head on outside in contact with the aqueous solution, hydrophobic tail on inside from core and forms barrier between cell and external environment. Look at here, hydrophilic region, hydrophobic region, hydrophilic region, phospholipid, protein, carbohydrate side chain. Now, cholesterol 6 hormone known as steroids, 4 fused carbon ring, different steroid created by the attacking different functional group to ring. This is the structure of cholesterol. Diversity in steroid, you can see the cortisol and aldocortisones, testosterone and estradiol and progesterone. So from cholesterol 6 hormones, what a big difference a little atom can make. Cholesterol is chemistry modified to produce the male and female sex hormone. You can see the estradiol is a female sex hormone and testosterone is a male sex hormone. This is the steroids. So important cell component, animal cell membrane, precursor of all other steroids, including vertebrate sex hormone, high level in blood, may contribute to cardiovascular disease. This is the cholesterol. Now, help keep cell membrane fluids and flexible. Now, protein. You know the protein structure, monomer, amino acid, 20 different amino acid, polymer, polypeptide. Protein can be one or more polypeptide chain folded and bonded together. Large and complex molecule, complex 3D shape. You can see the one amino acid, another amino acid forming polypeptide chain. Now, amino acid structure, central carbon, amino group, carboxyl groups, R group, side chain, variable groups confer unique chemical properties of the amino groups. So non-polar amino acid side chain, non-polar hydrophobic, you can see here. Why these non-polar and hydrophobic? Now polar amino acid side chain, polar or charged and hydrophilic. Why these polar and hydrophilic? Think about it. So amino acid with acidic side chain and their derivative, aspartate, glutamate, asparagine and glutamine. See this. Amino acid with basic side chain, you can see lysine, arginine and histidine. So building proteins, peptide bond, you know dehydration synthesis, linking NH2 of one amino acid to COOH group of another and forming CN bond. So look at the peptide bond. Now building protein, you know polypeptide chain, N-terminal NH2, C-terminal COOH, repeated sequence NCC is the polypeptide backbone, grow in one direction. Look at here, side chain, backbone, peptide bond, carboxyl end and C-terminal, amino N-terminal. So protein structure and function. Function depends on the structure. 3D structure, twisted, folded, coiled into the unique shape. Protein structure and function. Function depends on the structure. All start with the order of amino acid. What determine that order of amino acid? Lysozyme, enzyme is tears and mucus that kill bacteria. Now primary structure, you can see the order of amino acid in chain. Amino acid sequence determined by DNA. Slight change in amino acid sequence can affect protein structure and its function. Even just one amino acid change can make all the difference. So sickle cell anemia, you can see the valine, histidine, leucine, tyrosine, proline, glutamine. You can see the changing here in valine in place of glutamine. So normal red blood cell and the primary structure of normal hemoglobin, sickle red cell and primary structure of sickle cell hemoglobin. Now secondary structure, local folding, folding along short section of polypeptide, interaction between adjacent amino groups, hydrogen bond between R groups, alpha helix and beta belated sheet. Secondary structure, you can see the hydrogen bonding, electrostatic attractions, hydrophobic interactions, and metallations, coordinations. So tertiary structure, whole molecules folding, determined by the interaction between R group, hydrophobic interaction, effect of water in cell, anchored by disulfide bridges, hydrogen and ionic bonds. Look at here, hydrophobic interaction, clustering of hydrophobic groups away from water and van der Waals interaction, polypeptide backbone. So quaternary structure, join together more than one polypeptide chain, only then it is a functional protein. Collagen, skin and tendons. You can see polypeptide chains and hemoglobin. So protein structure review, you can see the primary structure, secondary structure, tertiary structure and quaternary structure. So capron in proteins, guide protein folding, provides shelter for folding polypeptide, keep the new proteins segregated from cytoplasmic influences. Look at here, cap and capsaicin, and see the polypeptide and correctly folded protein. So protein models, protein structure visualized by X-ray crystallography, extrapolating from amino and acid sequence, computing modeling, lysozyme. So denature a protein, disrupt 3-degree structure, pH, salt, temperature, unravel or denature protein, disrupts hydrogen bond, ionic bond and disulfide bridge. Some protein can return to their functional shape after denaturation, many cannot. So nucleic acid, you know the nucleic acid, what is the function, is store and transmit heredity information. Example, RNA ribose nucleic acid and DNA deoxyribonucleic acid. Structure, monomers and nucleotides. What is nucleotide? 3 parts, nitrogen base, carbon nitrogen ring, pentose sugar 5 carbon, ribose in RNA, deoxyribose in DNA and P4 groups. You can look at here structure. So types of nucleotides, 2 types of nucleotide, different nitrogen bases, purine, double ring nitrogen base, adenine and guanine, and pyrimidine, single ring nitrogen base, cytosine, thymine, uracil. You can see here purine, double ring system and single ring system. So building the polymers, pyrimidine, you can see the single ring, purine, double ring, and 2 sugars, ribose and deoxyribose. Here is the structure of polynucleotide. So nucleic polymers, backbone sugar to phosphate bond, phosphodiester bond, new base added to sugar of previous base, polymer grow in one direction. So N bases hang off the sugar phosphate backbone. Why is this important? So RNA and DNA, you know the RNA single nucleotide chain, DNA double nucleotide chain, N bases bond in pairs across the chain, spiraled in a double helix. Double helix first proposed as a structure of DNA in 1953 by James Watson and Francis Crick, and just celebrated 15th birth anniversary. So pairing of nucleotides. Nucleotides bond between DNA strand, hydrogen bond purine pyrimidine, double AT 2H bond, GC 3H bond. So why is this important? So informations polymers functions. Series of bases encodes informations like the letter of a book. Stored information is passed from parent to the offspring, need to copy accurately stored information, genes and genetic informations. So see the strand. So DNA molecule, double helix, hydrogen bond between bases join the two strand A, T and CG. Why is this important? Why that the strand are bonded by hydrogen bonds. So copying DNA, you know the replication, two strand of DNA helix are complementary. Have one can build other, have one can rebuild the whole. Why is this a good system? When in the life of cell does replicate occurs? So mitosis and meiosis. So DNA replications, it has not escaped our notice that the specific pairing have postulated immediately suggests a possible coping mechanism for the genetic materials. So Watson and Crick and others. Merrick Wilkinson. Now interesting note. Ratio of AT and GC affect stability of DNA molecules. 2 hydrogen bonds versus 3 hydrogen bonds. Biotic procedure. More GC need higher T degree to the separate strand. Higher T degree organize many GC. Parallelly, parasites many AT, don't know why. So another interesting note, ATP, adenosine triphosphate modified nucleotide, adenine ribose P1 means EPI, inorganic phosphorus, inorganic phosphorus. So macromolecules review. So carbohydrate, you know the structure monomers, monosaccharide, function, energy, new materials, energy storage, structural compound, example glucose, starch, cellulose, glycogen, glycosidic bond here. Lipids, structure building block, glycerol, fatty acid, cholesterol, hydrocarbon chains, functions, energy storage, membrane, hormones, example fat, phospholipids, steroids. You can see ester bond in fat. Protein, structure, monomers, amino acid, levels of structure, function, enzyme, defense, transport, structure, signal, receptors. Example, digestive enzymes, membrane channels, insulin, hormone, act. You can see the peptide bond. Now nucleic acid, structure, monomer, nucleotide, functions, what function, information storage and transfer, example DNA and RNA. So thank you. If my friends, if you like such type of video, then subscribe my channel and thanks for watching.